Hello, thank you for joining us for our webinar. This month, we are very pleased to have Dr. Sean Campagna with the University of Tennessee. Sean received his PhD from Princeton University, and he's currently an associate professor with the University of Tennessee in Knoxville. Sean is a renowned expert in the development and application of analytical techniques and multi-omics-based approaches to help us answer biological questions at the molecular level. So I'm really excited about the talk that we have today because Sean's going to be describing some state-of-the-art work that he has been doing with liquid chromatography and mass spectrometry-based metabolomics and lipidomics. He's going to discuss the new biomarker discoveries that have happened and show us how we can quantify thousands of molecules using these techniques that can then be used to probe the global metabolism of a sample. So it can really give us some in-depth knowledge in addition to what we already understand about these microbial populations. So it's a really nice complement to what you've heard from Microbial Insights in the past. So as many of you have attended before know that the webinar today is being recorded and it's going to be available on the resource section of the Microbial Insights website within the next week. We're also going to email out a link to all registrants as soon as it's available so that you guys can log on and listen to the webinar again. Now, at the end of this webinar, there's going to be a question and answer period. So please go ahead and type your questions into the GoToWebinar software as you have them for Sean. And at the end of the presentation, we'll go through and answer as many of those as we can for you. You can also email any additional questions that you have to us at info@microbe.com, and we'll get with Sean and get those answers for you and get back to you. So without any further ado, I'll go ahead and get started, and I'll turn it over to you, Sean.
Thank you, Joyce, for a kind introduction, and thank everyone for tuning in today. I'm really excited to have this opportunity to discuss metabolomics and lipidomics with you. So the talk today is actually going to start with an introduction to the technology because, as you notice in the slide here, it says developing tools, and I use the word developing both because we work on making new tools in this field, and also because the tool set is just now getting to the point where we can start to answer bigger and bigger questions with it. And we'll address sort of where we are in the state of the field, and we'll also talk about what we need to do better in this field as well to make technology, take technology to the next level. So first off, I'm going to give everyone sort of a little bit of introduction on what my laboratory thinks about, because we're chemists, and we collaborate with a number of wonderful people who help us on the biological side of things. And so I actually started my life as an organic synthetic chemist, and making small molecules, really looking at synthesizing bacterial metabolites. And then as a postdoc, I got very interested in bioanalytical chemistry, where we use liquid chromatography and at that time tandem mass spectrometry to try to measure all of the small molecules in a cell in a short amount of time. And
hey, Sean.
Really,
Sean, I'm sorry to interrupt. They're having a hard time hearing you. Would you be able to do it with a handset to see if that's clearer coming through?
Hold on one second.
Okay.
Figure out. Is that better?
That is so much better. Thank you very much. Sorry about that.
Okay, not a problem. And so, as I was saying, during my postdoc career, I got interested in bioanalytical chemistry, where the goal was to measure all of the small molecules in a biological sample. And when we were envisioning this technology, the reason an organic chemist got interested in that was because we started to notice that there were a lot more small molecules that make up living systems than anyone ever expected. And we'll address that today. And so today we use these two technologies, organic synthesis and bioanalytical chemistry, to try to map out metabolism and in a high throughput manner. And the systems where we primarily focus is on bacterial cell-cell signaling and metabolism. And really, we've started looking at that in the environment, and we'll address some of that today. Although near the end of the talk, we'll start talking about some current efforts that we have in more remediation and environmental monitoring type things, which hopefully will show you how the technology is developing into something that many, many researchers can take advantage of. So since this is a developing field, I'd like to really talk about what metabolomics is. And so metabolomics is really defined as a study of trying to find the chemical fingerprints that lead to phenotypes. And the world started with of metabolomics started with mapping out lipid metabolism, or lipidomics, where the idea was that we tried to measure the entire lipid soluble and lipid content of cells. And the reason that the technology started there was because lipids were based on known head groups with a combinatorial addition of a lot of tail groups, and it was just easier to build libraries and understand lipid metabolism. Since then, we've added the water-soluble molecules, which is everything else that you see in here, amino acid metabolism, sugar metabolism, TCA cycle, so all of the small molecules. And so today, really, metabolomics and lipidomics are starting to merge into a singular discipline where we're just trying to measure all of the small molecules in a cell. And I'm going to define that as anything with a molecular weight of 2,500 or under. So we're not talking about proteins or any of the large biopolymers. We're really only focused on sort of these metabolites and the inputs that go into biosynthesis and cellular metabolism. I'm going to use some jargon in today's talk, and we'll define that as we go along. The first of that is going to be the metabolome. And when I say that, I mean the collection of all metabolites, both water-soluble and lipid-soluble, that make up a cell, a biological system, or an organism. And then really what we're trying to do at the end of the day is we're trying to profile these metabolomes and ask the question, can we get any biological relevance from them? And metabolomics is actually just another addition to the systems biology set of tools that I think people are becoming more and more familiar with these days. We know that this started with the genomics revolution, where people could, for the first time, look at all the genes that were in an organism, which led to the ability to look at all of the RNA or the transcripts in the organism. Proteomics started to blossom, where people were trying to measure all the proteins. But what we found is that all of these technologies that measure genes, transcripts, and proteins really are technologies that happen before post-translational modification has taken effect to moderate or differentiate cellular function. And so we could really complement all of these studies by having a way to look at the actual state of the system or the physiology that's happening in the system, which is where metabolomics comes in. Because by measuring the small molecules, we can get an idea of how resources, energy, and is being flowed through a cell. And I often say that the small molecules really are the currency of life. And so we feel that this technology complements all of the other systems biology techniques that are really starting to take root today. And so when we talk about metabolomics, there are actually a couple of ways of doing the studies, and something that complicates the metabolomics field that you don't have in genomics and proteomics is the large chemical diversity in the metabolite pool that we look at. And I think we'll address that as we move forward in the slides here. So something else is sort of from a historical perspective that's interesting about metabolomics is when we think about all of these systems biology techniques that have been developed over time, they've sort of followed the central dogma of life. Genes go to proteins and proteins function to form physiology. And genomics was the first of the omics method. And this came about because Sanger's sequencing methods became available in the 70s or 80s. A massive expansion in mass spec technology led to the proteomics revolution in the 80s and 90s. Metabolomics as a word was coined in the early 90s to early 2000s. And I put a question mark there because actually Linus Pauling, probably one of the most famous chemists of all times, was interested in metabolomics in the late 60s and early 70s. And he actually was using GCFID technology to profile urine and breath metabolites, and thought that by looking at metabolism, we would actually have a window to view the differences in human genetics. And so even as early as the 1960s, chemists were starting to think, well, can we take small molecules and map those back to disease states? Unfortunately, metabolomics wasn't the first of the omics because analytical tools just weren't available to measure these large amounts of biomolecules and metabolites that were available. And so to give you an idea of the technology that Linus Pauling employed on the GC, he found 170 metabolites, and he was able to identify about 10 or 15 of those total metabolites out of the 170 that he detected. And so metabolomics lagged behind because the analytical tools to look at these very complex mixtures of biomolecules didn't exist. And Now, when we think about the metabolomics revolution, even though it's such a large analytical challenge where we know we're measuring tens of thousands, if not hundreds of thousands of molecules, if we can solve this problem, we really have a very, very powerful tool, because by looking at metabolism in a holistic way, we can answer questions on a scalable number of levels. And the metabolomics field started with the idea that we could differentiate systems by looking at entire pools of metabolites. This came out of the biomarker desire to find biomarkers for diseases from clinicians, where people started to ask the question, can we measure all of the metabolites and find a set of features, even if we don't know what they are, that differentiates the system and would tell us what state the system is in? And so this is where all of this started. But now over time, we've realized that if you're actually measuring every metabolite in a system and you're looking at tens of thousands of molecules, can we mine those data to get specific information on metabolic pathways, or even specific information on single metabolites as we move forward, right? And so this is where the field has come. And so if we can answer this analytical challenge of measuring all the small molecules, we should have a technology that would allow a lot of information to be gained and a lot of questions to be answered in a fairly high throughput way. Unfortunately, all of these things are very difficult. And so when we think about, sorry, I was checking for a slide there. When we think about how this has to happen, we really have to put a lot of effort into all of the analytical technologies that go into it, and which is really a challenge in the field that we'll discuss further in a little while. So with sort of that introduction of what we're trying to do and why it's important, I need to jump into a little bit more jargon before we jump into some biological vignettes here. And I just wanted to point out that there are really two types of metabolomics experiments. There's targeted metabolomics, where we're going to use tandem mass spectrometry to detect molecules that we know are there. So these are the standard pharmacokinetic type studies, or studies where you would ask the question, how much glutamate is in a system, where you really go in and look for a known set of exact molecules. What's great about these types of techniques is they're very, very quantitative. You know exactly how much of the compound you're detecting, but the metabolomics field has started to realize that we miss out on the ability for molecular discovery. And so, but if you know exactly what you're looking for, targeted methods are fast and quantitative. On the other hand, we also run experiments that we call untargeted metabolomics experiments. These are our broad screening experiments where we try to detect every single compound and metabolite in a sample. Unfortunately, the mass spectrometers and techniques that do this are only pseudo-quantitative at best. They're getting much better. So we can't exactly get exact quantitation for all of the molecules, but it allows us to look at many, many, many molecules in a sample, most of which we don't even know what they are at this point. And so if there's a mechanism of molecular discovery that needs to be had, these untargeted metabolomics techniques are the methods of choice. And I think it was surprising to people in the clinical world that there were so many biological molecules, even though anyone interested in the environmental world knows that dissolved organic carbon or carbon in soil or soil metabolites are very, very diverse. And so our laboratory actually focuses on both of these methods to move forward, and we'll talk about that in just a second. The other thing that jargon that's going to pop up in this talk is the idea of absolute quantitation. Most of the time when we measure metabolites, we're looking at comparisons between samples, and we just get relative quantitation or fold changes between the two. We just say sample A has more of metabolite X than sample B. There's an issue with getting absolute quantitation in that we either need to run calibration curves for every molecule in the sample, which is difficult if you're measuring tens of thousands of molecules, or we need to use stable isotope labeled standards, spike them in, and then get a ratio, internal ratio to determine exact quantitations. Unfortunately, that's very expensive to do. And so most of today's talk is going to focus on relative quantitation. Okay, there's one other thing that we want to discuss as well, and something that the field is beginning to find, is that the metabolite concentrations, or what I call pool sizes in a system, really are not quite enough to fully understand metabolic function, especially in microbial systems. And in fact, really what we'd like to know is the rate at which metabolites are being used or the flux through the system. And this is where mass spectrometric-based metabolomics actually has a really large value over some of the other metabolomics techniques like NMR and so on and so forth. Because if we can use stable isotopes, which can be differentiated by the mass spectrometer, because things that have carbon-13 are heavier than things that have carbon-12, and things that have nitrogen-15 are heavier than things that have nitrogen-14, and we can sample over time, we can actually set up a set of standard differential equations, sample over time, and measure biological fluxes and rates through a system. Now, I promise you that I will not get into all of this math, and you will never see a differential equation again on these slides, but I just want you to be aware that these techniques are being used in the system. And the way that they work is actually to take a sample that's grown on normal nutrient sources, to somehow introduce an isotope media that has carbon-13 or nitrogen-15, and to sample over time in the system, and then look for these metabolites that start out as an endogenous light metabolite with carbon-12 or nitrogen-14, watch the light metabolite disappear, which tells you the rate at which things are being used, and then to watch the metabolites with heavier stable isotopes appear, which tells you the rate at which things are being biosynthesized. And so by looking at the disappearance and the biosynthesis rates, we can actually get a flux through a pathway, and we can start to answer questions on whether or not a metabolite is being depleted because of lack of use or lack of biosynthesis, and so on and so forth. And this is a very powerful way for us to actually get into monitoring metabolism at sort of the next level above concentrations. So that being said, this is what our platform for metabolomics looks like here at UTK, and I think this is fairly representative of the platforms that happen. And I'm also going to use this as an ability or an opportunity to tell you about how this technology has developed over the last 10 or so years. So when I started here at the University of Tennessee, we had an HPLC triple quadrupole instrument that was doing targeted metabolomics, because at the time the field really was focused on the 700 or so metabolites that you find in your standard biochemistry textbook. Of those 700 molecules, about 400 of them could be purchased. And so the standard metabolomics platforms at the time would buy those 400 molecules, set up detection parameters based on tandem mass spectrometry for those molecules, and they would run the samples through these mass spectrometers looking for basically one molecule at a time in an iterative process. And most people were measuring 30 to 40 molecules out of biological samples. We had brought the method that we had developed when I was a postdoc at Princeton. We were measuring 120 known water-soluble compounds and could perform these flux analyses in about 2 hours using our targeted metabolomics methods. And we were ecstatic because we could answer a lot of biological questions with that. About that time, Thermo Fisher Scientific now, introduced this mass spectrometer called the Orbitrap, which is a very high resolution mass spectrometer which could ID compounds based on high accuracy molecular weight, which allowed us not to do tandem mass spectrometry and just do standard mass spectrometry identified by mass alone, which allowed us to look at hundreds of molecules in a scan versus one molecule in a scan. I'll answer questions about that in the end if people have more technology-based questions there. But what that allowed us to do was to start doing untargeted metabolomics along with looking for the metabolites that were in our library of compounds. And so our library didn't expand beyond 400 molecules because that's really all that's commercially available when we look at the water-soluble metabolome. But what we and everyone else in the field started to realize is that there were thousands of biological molecules, which we'll call spectral features, from compounds that we knew were real metabolites, but we still to this day don't know their structure. On top of that, since we were measuring hundreds of compounds per scan, our scan rates, our sampling time went way down. And so a couple years ago when I made this slide, just by using this Orbitrap instrument, we were able to measure these 150 known water-soluble compounds and replicate everything that the triple quadrupole was doing. But we're also measuring 8,000 or so spectral features from every sample for molecules that we knew had were from biological sources. Many of the times were important for the phenotype, but still had an uncharacterized structure. These days, with some developments in the method and more powerful mass spectrometers, this number is skyrocketed from 8,000 to about 24,000 molecules per sample. We also started complementing our metabolomics measurement with a lipid measurement. The library was larger because lipids are similar to each other in lipid classes. And here, again, we were measuring tens of thousands of molecules, closer to 30,000 these days from our lipid-soluble analysis. And so now in the period of just a decade, we've gone from measuring 120 molecules to measuring about 50,000 molecules per sample in 1.5 hours of real time and 2 hours of instrument time. And we complement that with a lot of other technology development. And these days, we're also complementing that with some limited proteomics and cross-omics analyses as well. But what we found as a field in the metabolomics world is that we're at a point where we can provide a wealth of information to biological researchers. And what's interesting as an organic chemist, and coming back to that, is even though we're measuring 50,000 molecules, you'll note we're only identifying about 500. And so just the diversity of compounds and the wealth of information that is left to be learned about metabolism in general is both staggering and very exciting for people who are interested in how small molecules function and pattern the world around us in the biological arena. So that is the technology that we're going to employ in this for the rest of the talk. And now we're going to shift from talking about technology and analytical chemistry and sort of the high level overview and tell you how we've used this technology to go in and look at a number of environmentally relevant biological systems. The first one we're going to tell you about is a microbial system that was taken from the photosynthetic layer of the world's oceans and really was trying to help answer the question how viral lysis patterns the movement of nutrients through the oceans. So surprisingly, it wasn't until 1999 that Wilhelm and Suttle discovered that microbial viruses were a major contributor to mortality in the top 5 meters of the world's oceans. And in fact, viral lysis probably leads to 30 to 50% of all microbial organisms dying per day in the world's oceans. And so when we look at these food webs or photosynthetic organisms take sunlight to make sugars to feed heterotrophic bacteria, it was thought that most of that biomass from the heterotrophic bacteria was either taken through protists or other things that ate the bacteria all the way up to zooplankton, or that through some mechanisms of lysis, there would be some release of these metabolites into dissolved organic matter and dissolved organic phosphorus matter. What we now know is that viral lysis also plays a major component here. And so if 30 to 50% of these bacteria are dying on a daily basis, it means that there are a large number of metabolites and nutrient sources that are being released into the world's oceans that have never made it into any models of the food web. And so this study was with Allison Buchan and Steve Wilhelm here at the University of Tennessee to try to understand whether or not we could quantitate the effects of viral lysis on microbial populations. When we started this study, initially people told us that there was no reason to look at bacteriophages or viruses in these organisms because everyone knew how phages and E. coli worked, and therefore we knew how all bacteriophages worked, which is interesting, and I'll point out sort of a fallacy of using model systems is that they often can't be taken to environments where the model doesn't hold. And so we started with the idea that when we looked at these coliphages that affect E. coli, all of them seemed to work the same, that the viruses would enter the host, they would take over host DNA synthesis, and really just turn the E. coli cell into a microbial factory, or excuse me, a virus factory, and lyse the population of E. coli cells, and then the viruses would need to find new hosts. A lot of these viruses contain auxiliary metabolic genes to do this, and to help the host overcome the rate-limiting steps in virus synthesis. And this was really the entire view of what bacterial phages did at the time. Now, about 2011 or so, we started about this study, people started looking at phages in the world's oceans, and they were starting to find that the phages, again, had these auxiliary metabolic genes where they could help boost photosynthesis rates, or that they had other systems for helping to sequester resources. And so that sort of solidified a lot of people's biases that we knew everything we needed because of the E. coli phages. But finally, we were able to convince some people to let us look at a phage that infected a Rosiobacter strain that was taken from a mesocosm in the world's oceans right outside of Bergen. Now, Rosiobacter are interesting because they're numerically dominant in near coastal populations and are probably responsible for a lot of the turnover of nutrients in those systems. And so Alison Buchan, our collaborator, isolated this Rosiobacter. And what was intriguing is that the Rosiobacter came with a phage, Rosiobacter 2047, Rosiophage 2047, A and B already in the genome. And so this Rosiobacter does not exist in the environment without a lysogenized phage in the genome, and there's a lytic complement that goes with that. And so initially, Alison was interested in looking at these systems because if you look at them over time, what you find is that uninfected cells grow in biomass. You can see here, if you estimate the total carbon in femtograms per milliliter, that yes, your need for carbon goes up, or your need for carbon goes up if you're just a bacterium. But if you have an active viral infection where you stimulated the lysogenic phages to become lytic, the population, even though the bacterial population goes down, much more carbon is shown here, and the black bar is sequestered in viral particles. And so there's much more nutrient turnover in the environment in the presence of active viral lysis or viral populations. And we were interested in what impacts that had in these communities of bacteria as based on the model system in Rosiobacter. And so we worked with Alison to actually perform a metabolomics experiment. Sorry, the slide's moving slowly here, where we looked at the metabolome of this infection cycle through the entire course of the infection. And if you look at what happens to the microbial population, as shown here in the dotted line with the circles, the microbial population grows normally until about 240 minutes post-infection. Then viral lysis is noted, and the microbial population crashes from its peak to about 30% of what it was at the top. If you look at the phage population based on gene copies, you can see that for the first 60 minutes, there is an elevation in the number of phage after addition of some of these lytic phages to induce the cycle. Then from 60 minutes to 240, you have this rapid progression where you see a large number of phages being generated, lysis occurs, phages are released, and then the infection cycle starts over again. We performed a metabolomics experiment where we looked at all of those small water-soluble molecules, in this case in a targeted fashion, and at the time point shown here. And at the black bars, we also performed a flux experiment where we added carbon-13 labeled nutrient sources to look at the turnover rates of the nutrients in the system. So we looked at that before viral lysis happened, and then at the peak of viral lysis. And what we found was that in contrast to coli phages, where you expect cellular metabolism to dramatically change at the beginning of the infection cycle, that in this case, cellular metabolism is monitored by the percent of metabolites that show statistically different changes, really isn't altered until lysis occurs. And when lysis occurs, you can see that from between 10 and 20% of the metabolites that were measured being different, we now see 80 or so % of the metabolites to be dramatically different in intracellular metabolism. This was initially intriguing because it was unexpected, right? We expected certain pathways to be dramatically altered in the beginning, which would lead to an alteration, of course, the entire infection cycle if it were working the same as E. coli, which in this case, it looks like it wasn't. So when we go and look into these metabolites even further, again, the graph of the infection cycle is shown here. What we saw was that when you looked at the intracellular metabolites, and we'll talk about what the data format means here in just a second, that the cells that were infected end up having a very high concentration of metabolites at the end. And from here on out in the talk, we're going to use this format that we call a heat map format to show these large number of data. And I don't want people to get hung up on actually trying to read the names. That's not the point. These are to show qualitative trends on how things change. And each one of these cells, so for instance, this bright red one here, is really a measurement where we've taken the intracellular metabolites in the experimental population, in this case, the populations of cells that have a viral infection, and we compare that to the control population at the same time, so the cells that had no viral infection. And then we just form a ratio. If the ratio leads to a measurement being higher in the infected population, we color it red like it's hot or on fire. And if it's lower in the infected population, we color it blue like it's hot, cold. And that's why we call it a heat map. So for instance, this cell here at 60 minutes means that we have higher succinyl-CoA in the infected population than we do in the uninfected population of viral cells in this case. And so as you can see, this leads to the ability to note that over time, the infected cells actually had a large number of metabolites that were higher in concentration. This is on the log2 scale, so saturation is an 8-fold increase. In most of the cases, we saw a 6 to 8-fold increase in metabolite concentrations in the infected cells. That was intriguing to us at the time because we actually didn't know if that increase in concentration was because the cells were sick and metabolism had slowed down, and so things were just hanging around in the cells, or whether the cells had become where the metabolic activity was due to sort of a hypermetabolic state where they were churning through a large number of metabolites. And a failure of metabolomics where you just measure concentrations is that you will never know that, whether or not a buildup in something is due to a lack of utilization of downstream metabolites, or whether or not it's used to increase, or whether or not it's due to increased biosynthesis. This will become important when we talk about the flux studies on the next slide, because what we saw when we looked at the flux studies, we were able to go in and look at these two differences. Okay. So when we looked early in the infection cycle, we really saw no differences in fluxes. However, when we looked late in the infection cycle, what we saw was that the turnover rates seem to be the same in the two, and we'll talk about how this graph works. So here what you see is the blue is the uninfected culture. You can see the disappearance of the unlabeled metabolite and the synthesis of the C13 labeled metabolite. So it's really the slope of this curve that tells you the turnover rate. And for most of the metabolites in there, both the infected and the uninfected cells were using their metabolic pool at the same rate, and they were resynthesizing their metabolic pool at the same rate. This doesn't look like it's any useful data until you realize that all chemical rate equations actually have two terms. They have the rate term, and then they have the concentration term. So for all of these metabolites in the infected population, where you have 4 to 8 times the concentration that's being used at the same rate, you actually have 4 to 8 times higher flux or metabolism through all of these pathways. So interestingly, these Rosiobacter look like they had entered a hypermetabolic state due to viral infection. This trend actually held true for all metabolites except for 2. And if you look at glutamine and glutamate, which are the two that were different, you can see that the infected cells were using their glutamine pool more slowly and using their glutamate pool more slowly, as evidenced by both the slower disappearance and the slower rate of resynthesis in these two metabolites. Now that was intriguing because glutamine and glutamate are the primary ways in which cells assimilate nitrogen and ammonia. And we know that in these environments, nitrogen tends to become a limiting metabolite. So we started thinking more about this and asking the question, could we get more information out of this? And so we noted that the glutamate to glutamine ratio is actually a measurement on nitrogen status in systems. Glutamine holds 2 nitrogens, glutamate holds 1 nitrogen. So as you become nitrogen starved, you tend to have less glutamine in relationship to glutamate because you're taking the nitrogen off the amide on the side chain and using it for other biosynthetic processes in the cell. And so in the uninfected population, what you see is that the glutamate to glutamine ratio increases over the time of the experiment, indicating that we're entering a regime of nitrogen limitation to where the infected cells actually are maintaining their glutamate to glutamine ratio, indicating that nitrogen is not going to be a problem in these cultures. They're maintaining enough nitrogen to function. Now, when you ask about what all of these data mean together, what it shows is we have a system here where viral lysis is actually re-releasing nitrogen to the environment and allowing these cells to maintain their nitrogen status in a regime where nitrogen is limiting. On top of that, it also should be noted that these cells biochemically are much different than E. coli. We all know that E. coli goes through dioxide growth, that if you give it 5 different sugars, it eats sugar 1 before it eats sugar 2, and so on and so forth, to where the Rosiobacter are able to churn through everything very, very rapidly. And so this gave us a picture where these bacteria that live in sort of a feast and famine environment have the ability to utilize all cellular inputs very, very rapidly and seem to have a relationship where the virus is able to be triggered for a variety of unknown reasons. But when it does, cellular mortality of kin virus is actually beneficial to the viral population that is there. They can keep nutrient concentrations high, both in high value nutrients that contain nitrogen and also in total nutrient population through I don't want to say symbiotic or synergistic relationship because those have real biological meaning. But here, this isn't a case where the virus seems to be net detrimental to the system. It actually seems to have a real function in nutrient turnover in the systems. So this is sort of our first indication as a laboratory that we could go in and start answering real environmental questions using metabolomics, and also was intriguing for us because it told us that the world is a lot more complex and a lot more beautiful in systems than we can learn just by looking at coli viruses in the laboratory. So from there, we've moved on to a few other studies. And again, I apologize, my computer is moving very slowly. From this, where we've gone from sort of doing this targeted technology now to employing our untargeted technology. And in the essence of time, I'm actually going to skip this section here and talk about our maneuvering into looking at metabolism in a soil system using our new techniques. And my plan is to talk about another 10 minutes or so, then we'll open this to questions. And so this was actually done after I brought Dr. Hector Castro into the laboratory, and the reason that this slide actually has a full citation because these are unpublished data. This is done in collaboration with Amy Klassen and Michael Strickland, who have been really great in helping us move into this arena. And so the goal of this study was to ask the question, could we use metabolomics to start looking into ecology in the soil in the same way we've done in the world's oceans? And the first thing we found was that soils were much more complex than seawater, right? They're a heterogeneous mix of things, different types of soils, different types of particles, different types of organisms, plant material. And so Hector actually spent a lot of time trying to figure out how to extract metabolites from this. As you can see, most of it is mineral particles and water, and we're really trying to get organic matter out of a very small fraction from this. And so it's to his credit that he was able to develop techniques where we can get usable data out of several hundred milligrams of soil and actually measure many, many molecules. And so after going through sort of extraction protocols and optimizations, we started looking at the metabolome in these systems. And the first study we did was actually to ask the question, well, what we needed to go through and understand on the metabolomics end. We knew we had the mass spectrometer that would function. We knew people had done genomics and proteomics, but again, we were very worried about whether or not we were going to get enough small molecules out of the system. And so we partnered with Michael Strickland first to actually and receive soils from a large soil management study where he was comparing different top growth and different plants that had been placed on top of lands as a mechanism to keep soils intact. I am not a soil management person, so I probably totally butchered the design of the study. But really for us, we were asking the question, how does cultivation, mixed hardwoods and forest change soil metabolomes over pastures? And as you can see in this, there's a much different amount of biomass in each of these soils when we collected them. And these are data that Michael sent us. And he also noted that the relative respiration rates were very different in these different soils, and that the concentration or respiration based on these different molecules was different. And so we performed metabolomics on all the soils and asked the question, could we start to fingerprint them and understand other differences in them? When we looked at either 30 milligrams of soil or 100 milligrams of soils, you can clearly see that when compared to pasture, you can start to find differences where the mixed hardwood had a lot more of these nitrogen containing and TCA cycle, carbon utilizing metabolites than did the pasture. And there's some other patterns in there and so on and so forth. What was sort of intriguing to us was that we actually weren't seeing a large number of known metabolites. But even though we were seeing these known metabolites in soils, we were able to see differences in the populations. We think we're not seeing a lot of known metabolites because many of them are going to be utilized very rapidly by the bacteria, which are at relatively low concentrations compared to samples that we normally get. But again, we still were seeing differences in these soils and would be able to tell what type of soil we had just by looking at these metabolite profiles. From there, we performed principal component analyses that look at variation vectors between the soils. And again, you can start to see some separation just using the known metabolites, with mixed hardwood being the one that is different, most different from all of the other soils, again, because of those nitrogen and TCA metabolites that we see there. Now, while this is good, separation here isn't great. And so Hector went in and asked the question, if we're measuring all of these unknown metabolites, where we don't know their structure, but we know they're from biological sources, would those give us more information? And so if we actually move to looking at these soil metabolites, of which we detected 4,000, roughly 5,000 of these soil metabolites, even though we don't know what they are, you can see that the biomarkers or metabolic signatures of these soils now become dramatically different. It would be very easy to classify any of these soils based on these biomarkers of these unknown molecules. And in fact, we see almost 8,000 significant differences among these populations. And we see 10,000 fold changes where there's at least 50% more of one of these molecules in one of these populations. And so what we've now begun to find out in these systems is it's really the unidentified spectral features, all of these biological molecules, biologically derived molecules, that we have no idea what they are, that are really probably important for the differences in physiology and the difference of states in these systems. And so we're currently working to identify as many of these as possible. I can tell you as an organic chemist where molecules are synthesized at the rate of, 1 or 2 per year, we're probably not going to make it through 4,800 anytime soon without a large influx of organic chemists and a large influx of funds. But I would also note that in these systems, even though we don't know what they are, we do know that they're driving differences. And what's nice is if we start to believe that a small subset of these are causal, we can go in sort of a directed way and identify these features one at a time. And I think it really gives us the opportunity to learn a lot about biological systems. And I think it will become increasingly important. And in fact, when I started in the metabolomics field, it really was with the idea of finding out these unknown features. So I'm going to leave everyone with talking about some research that has nothing to do with environmentally relevant systems, but sort of highlights the kind of experiments that are going to be needed to map out this sort of unexplored space in metabolism. This was also the last study that I ever did hands-on myself without the help of numerous very good students, even though my student Jessica Gooding also helped with this when we moved to Tennessee. And this was an experiment where we were asking the question, if metabolic pathways are like a assembly line where A goes to B, goes to C, and goes to D, and we knocked out part of that machinery in the assembly line, we should see substrates build up in the mutant, and we should see products disappear in the mutant. So if we could rapidly collect metabolomes of wild-type organisms and mutant organisms, we should be able to find substrate-product pairs and to map those back to the genomes and the genes of unknown function. And so that's exactly what we did. We started going through the yeast magic marker deletion set for all of these genes of unknown function, collecting their metabolome and asking the questions, did we see sort of this pattern where one metabolite was up in concentration and the other metabolite disappeared? And after going through about 100 or so organisms, we found one where, the concentration of the metabolite shown here was way up, and other metabolite are way up in the mutant and not in the wild type organism. After some work to characterize the structure, the research team found that this molecule was cetoheptulose 1-phosphate, cetoheptulose bisphosphate, which are 7 carbon sugars. Octulose 1-phosphate, octulose bisphosphate were also up in concentration, and octulose 8-phosphate was down in concentration. So again, we saw things that were up and things that were down. Now, initially we were confused about this because if you go back and look at your biochemistry textbook, what you find is that 7 carbon sugars are there only in photosynthetic organisms, and the rest of these sugars aren't even annotated in the genomes or metabolomes of organisms out there. And that's because what people knew about sugar metabolism at the time was that 6 carbon sugars, each carbon here is shown as a ball, either went through the oxidative pentose phosphate pathway to make ribulose 5-phosphate, which is needed for DNA synthesis, or they went through glycolysis to be chopped up for energy. And people knew that 7 and 8 carbon sugars were in cells, but what they thought was all the aldolases down here were making them by mistake. And through a lot of work that I'm not going to talk about today, what we found is that the enzyme that was encoded by the gene that was knocked out in this organism was cetoheptulose bisphosphatase, actually was a thermodynamically committed enzyme that took cetoheptulose bisphosphate to cetoheptulose 7-phosphate. Once that occurred, these aldolases can no longer function. And so it trapped this 7-carbon molecule in sort of a state where it was unclear that the cell could use it. This is where some high-level bioinformatics by Klasklin and Caudy came in, and they determined that yeast actually have an entire biochemical pathway to take cetoheptulose 7-phosphate all the way to ribulose phosphate and contain a non-oxidative pentose phosphate pathway. That was intriguing because it was the first fully described biochemical pathway in yeast in a couple of decades. At the basic science level, what is also intriguing in this case is that if you can make DNA and not change your oxidation state, you can actually overcome growth problems in a number of conditions. And we all know that Yeast make a number of wonderful products for people, most of them small alcohols, either for biofuels or recreational purposes. And one of the reasons that that is limited is because of the oxidative load of fermentation. And so if you can reroute around that, we could potentially use this pathway to engineer yeast that could produce higher levels of fuels. It's also turned out that people now believe that human cancer cells or oxygen is limited also use a non-oxidative pentose phosphate pathway, which just shows that as we describe more and more biochemical pathways, we learn more and more about the world around us and how physiology functions. So with that, I'm just going to throw up the acknowledgement slide. None of this would have been possible without a number of wonderful students in my group, many of whom are starting to move on to their own independent careers, collaborators in studies that we haven't talked about today, and then collaborators highlighted here had an integral part in studies that were talked about today, plus a number of other people who really helped us enrich our scientific community in the metabolomics world by giving us interesting problems to work on. And then these are the organizations and agencies who have funded this work, and we'd like to thank them as well. And with that, I'll be glad to answer any of your questions.
Great. Thank you very much, Sean. That was a great presentation, a lot of information for us to think about since this is kind of new to a lot of us doing remediation type work. So one of the things that came to my mind immediately when you were talking is, have you looked at any metabolites that are specific to like anaerobic degradation pathways, for instance, benzene and naphthalene? I know that's something that we haven't quite elucidated which genes are important, so we often have a hard time documenting degradation anaerobically of those compounds. So I wanted to see if you've done anything with that and kind of what you've seen as far as results.
We have not done anything with that, actually. Most of our work is focused on understanding sort of the photosynthetic layer of the ocean, but we would love to do that. I think we're just now in a regime where we can look at systems that are more complex with soils and other biological particles in them. So yeah, I'd love to talk with you more about that, actually. So.
Okay. Yeah. So I guess if there's any projects that people have that are interested in that, maybe that's something we could talk about, ways to kind of examine what metabolites may be important for that degradation.
Right. Yeah. I think that if we sort of relate it back to the soil management project, we're at a point now where we can start asking the question, are there biological signatures that could tell us something about what's going on in the systems? And I think the answer is yes. And so I think what's nice about metabolomics is it's definitely a platform that can be applied to a lot of things, right? It sort of doesn't care where the sample comes from as long as there's enough biological material to measure. So.
Right, and you can tend to get pretty low concentrations, correct, when you're looking at these metabolomic approaches?
Yes, yeah. We not only just low concentrations, but a very broad difference in concentration. So we measure metabolites that are in the micromolar range all the way to metabolites that are, in the nanomolar range as well. And so we get that dynamic range of concentration measurements. What's also nice is we've actually used samples that have very, very low cell densities and very, very precious samples. So one of the studies that's coming out that I didn't highlight today with some researchers here on campus is we've been looking at organisms that were collected 80 meters below the seafloor in these boring projects. Boring as in taking cores out of things, not boring as an uninteresting project, where people are looking at sort of these organisms that have very slow metabolic rates because they're under very high pressures and very low temperatures. Some of these ancient organisms that are persisters, and we've been able to get metabolites out of soil samples that probably have a couple hundred active bacteria per microgram of sample or so. I don't even know if we know the cell densities down there, but we're able to measure metabolites from there. It's not 50,000, it's 700-800, but we can work with just about anything that comes to us.
Great. All right, one question that we had submitted says, have you utilized spectrophotometry as a non-targeted metabolomic approach to assess the metabolome of the aqueous system? And if you've done it, have you found any relationship between the spectrofluorescence and the metabolomic process itself?
So we have not, and I guess I should state that we focus on chromatography, mass spectrometry, metabolomics. NMR metabolomics has actually got a fairly large following as well, because it's very quantitative for the measurement of about 40 to 50 metabolites per sample. And sort of the photometric methods are also out there. We actually have a researcher, Frank Vogt, here in the department who uses Raman to look at signatures, and he can definitely differentiate different types of blooms of algae and things. And we've been discussing with him how to sort of merge the two technologies, but we have not yet done it, even though it should be done, because metabolomics works better the more metadata and other data that you have. And really the true power of any of these is cross-omics, because we're all answering a small part of the study. And so, yes, I would love to look into the correlation there, because one is far more rapid than the other. And I think they both tell you very different information. So I don't know if that answers the question. Yeah.
I think so, and I think it would be interesting. If you have a moment, I wouldn't mind you expanding a little bit. We talked earlier about some of the work that you're doing looking at reductive dechlorination and the projects there, and I think that's something that a lot of our audience has interest in. So can you talk a little bit about the crossover that's happening between using, routine qPCR data that's kind of become the gold standard for understanding remediation, looking at the dihalococoides concentrations. And then do you see the metabolites being a nice complement to that we could, better understand rates of degradation and kind of maybe how that'll all fit together with our expanded knowledge as we move into doing more proteomics of these cells?
Right. And I think I'm actually going to try to head back to a slide here so I can point at things if I can get to it. I apologize to everyone for this. Right. So really the way these technologies, I see them working together, is if one can go in and look at either transcripts or genes through qPCR, what that really tells you is that the potential is there, right? There are organisms that have the gene in the environment. And if you look at the proteins, you could then understand whether or not the protein to perform the function is being expressed. What the metabolomics allows you to do is actually look at the products of the protein or the products of the metabolism and answer the question, are the proteins functional, right? Have they been turned on? Are there enough of them there to actually make a difference in things? And so that's really the way it all complements, right? The genes and the transcripts tell you that their potential is there. The proteins tell you that the organism's trying to utilize the potential by building the machinery to run the process. And the metabolomics tells you the extent to which the process is running. And so that's the way I see all of these things coming together. They're all different measures of activity that give you a little different view on different parts of the process. So, and I think when you bring them in combination, they all have much more power than if you look at any of them in isolation, right? So.
Do you feel like you've identified some metabolites that'll be important for reductive dechlorination?
We actually have grown the cultures and have not run them yet.
Okay.
So we are heading that direction, but at this point we don't have anything other than some very, very preliminary data. So, but maybe we can do a follow-up once that study's done.
Yeah, definitely. I think that's of a lot of interest. The other thing I had a question on, one last question before we let you go today is, you mentioned some work that you've done looking at bacteriophages. And have you done any work with bacteriophages in degrader populations at contaminated sites? And if you have, I'd be curious to see your experience if you see that, there's any inhibition or limitations that happen if you have a lot of bacteriophages that are present.
No, I mean, we haven't actually never thought of it until now. That would, again, be a very interesting study to see how the organisms there sort of alter things, right?
Yeah, I'd be curious.
One of the problems that
I'm sorry, I was just going to say, I've been curious to see if they affect specific bacterial hosts or if you see differences with different microorganisms and how they respond with the phage.
Right. Yeah, that question might actually be better answered by qPCR in the communities as a whole than metabolomics. But if there was an isolated phage microbial system, it would be intriguing to see how the phage affected the metabolism of that single microbe. So, one of the things we found in all of these degrading systems is they grow so slowly, right? That we've had issues gaining enough biomass and enough samples to go in and really look at things in sort of the same high throughput manner that we've done in oceanographic systems, where the doubling time of those Rhoseobacter is 40 minutes or so. The helicocoides and some of these organisms double on the rates of days, if not longer. So, there is that real technical issue with following them in longitudinal studies, and hopefully we'll overcome that soon and have some ways to look at them.
Well, I'm really excited to see what work comes out of your lab, especially as you're working on some of these remediation type projects, and we look forward to working with you more on it. So thank you very much for your time today and sharing with us. We'll definitely have you back in the future as you dive deeper into some of these analysis that you're doing. So thank you very much, Sean, for your time. We appreciate it.
I appreciate that, and thanks to everyone who listened, and it's been a great opportunity to kind of share what we do. So thanks again.
All right. Thank you, Sean. Thank you, everyone, for attending. And we'll have the webinar available on our website within the next couple of days. And if you have additional questions, send them to us at info@microbe.com, or you can email them to Sean, and we'll be happy to answer them. All right. Thank you again, Sean.
Hey, thank you. Bye-bye.
All right. Bye-bye.